Hello everybody, a very good afternoon to all of you. In our previous video of recombinant DNA technology, we have discussed up to selection and isolation of DNA. Okay. In this video, we will discuss about the DNA which can be obtained from mRNA and the detail about restriction endonuclease enzyme. Okay. In our previous video, we have discussed that the gene of interest can be directly obtained from the DNA. Okay. Suppose in certain cases the DNA is not available. In that case, we can select the gene from the mRNA. So, let us discuss about obtaining DNA from mRNA, how it is possible. The messenger RNA, for this process, the mRNA first fractionated from the cell through gel electrophoresis. mRNA obtained by gel electrophoresis. Okay. After obtaining mRNA, what happened? It is treated with enzyme reverse transcriptase along with deoxyribonucleotides. Okay. This mRNA treated with mRNA treated with reverse transcriptase enzyme, reverse transcriptase along with reverse transcriptase, the deoxyribonucleotides are used. Okay. By treatment of this enzyme, another complementary DNA strand obtained. This complementary DNA can be isolated by treatment of alkaline phosphatase. Okay, by alkaline treatment, the complementary DNA can be. This complementary DNA after isolation is again treated with DNA polymerase 3 enzyme, DNA polymerase and the same nucleotides, deoxyribonucleotides primer. These are used to obtain the DNA or segment of gene. This is the method by which we can obtain the gene of interest in some cases where the DNA is not available. Let us discuss about the enzymes which are used in recombinant DNA technology. Okay. So, we have selected the gene of interest either directly from DNA or from mRNA through the process of reverse transcription. Okay. Then come to the selection and isolation of enzymes. A variety of enzymes are used in the process. Let us discuss about the endonuclease enzymes first. After that, we will discuss about other enzymes. Okay. The enzymes are basically essential to cut the DNA segment at the desired location. Okay. The enzymes which are used to cut the DNA, suppose this is the segment of DNA and this is the gene of interest. When the enzymes used to cut the DNA, these are called as endonuclease enzymes. The endonuclease which are used to cut the desired site or restriction site are called as restriction endonuclease. Okay. These restriction endonuclease are specific group of enzymes which identify 4 to 8 bases as the recognition site. These are also called as molecular seizures. They cut the DNA at a specific location. Okay. The cutting is due to splitting of phosphodiester linkage. Okay. After splitting of phosphodiester linkage, there is a 5' phosphate and 3' OH. At 3 position 5 prime position, phosphate group, a free phosphate group is present. At the 3 prime position, there is a OH group. Okay, this is the resulting DNA which is isolated after endonuclease treatment with the desired DNA. Okay, these endonuclease enzymes are categorized into 3 groups. What are the characters of 3 groups of enzymes? Many endonucleases are available from the host cell, that is from prokaryotic cell, which are classified into 3 varieties or 3 groups. The first enzymes are ECO R1. These are group 1 enzymes. The enzymes are named according to nomenclature rule. The naming is according to generic and specific name of the host, that is from E. coli. 1 letter from genus, 2 from species, and the type 1, for type 1 we have written R1. Okay. This ECO R1 is formed of 3 identical subunits. 3 identical subunits used in formation of enzyme. The molecular weight is about 4 lakh Dalton. Molecular weight is about 4 lakh Dalton. This enzyme have endonuclease and methylase activity. Endonuclease it can cut, and methylase it is responsible for methylase activity. These 2 activities confined to type 1 enzymes. It got the site of location, that is 1000 nucleotide away. It got the restriction site 1000 nucleotide away from the restriction site. Okay, this is all about E. coli R1, ECO R1. Okay, next to ECO R2. ECO R2. This enzyme is one of the most suitable enzyme that is used in the process of recombinant DNA technology. Why? Because this E. coli R2 is formed of 2 identical subunits. The molecular weight varies from 20,000 to 1 lakh Dalton. It has a recognition site of 4 to 6 base pair long recognition site. It always recognize palindromic sites. Okay, palindromic sites means, suppose this is one strand having the sequence of bases while the other strand have the same sequences in opposite, in reverse direction. Okay, this is the palindrome. It recognize the palindromic site for cleavage. Some of the enzyme generate blunt end while some other enzyme produces sticky ends, because there is a axis of symmetry which this axis of symmetry divide the segment or palindromic side into two identical units. Okay, this is the axis of symmetry. It divides the palindromic sides into two identical parts. The enzyme which cut the DNA along the axis generate blunt end, while some other cut the DNA on either side of axis, which produces sticky end or cohesive end. Let's discuss about the blunt end and sticky end produced by EcoR2. Okay. Suppose this is a recognition site, this is another recognition site. Remember, always the EcoR2, it require magnesium ion, as a cofactor for its action. When this enzyme cleave the DNA along the line, along the axis of symmetry, it always produce, suppose this is the axis of symmetry, it always produce a blunt end. Okay, this suppose the phosphodiester bond break on, it generate blunt ends like this. These are the blunt ends, T, G, G, A, C, C, another C, C, A, G, G, T. This became the blunt end. When EcoR2 enzyme is used, it cut the DNA on either side of symmetrical axis. EcoR2 cut DNA at location G. So here G, C, T, T, A. Another location site here G, C, T, T, A. Okay, these are the palindrome. These are the palindromes which cut the DNA producing. This is the sticky or cohesive end. This is the blunt end. This is sticky or cohesive end. These two type of cutting are generated by endonuclease enzyme. Okay, so it is the most suitable end which help in complementary base pairing during annealing process. Okay. Then come to EcoR3 enzyme. It is all about EcoR2 type 2 enzymes. Then next EcoR3 enzymes. The R3 enzymes are, these R3 enzymes are another group of enzymes which are formed of two identical subunits. But what is the difference? It cut the DNA, cleave the restriction site 24 to 26 base pair away from this site, from the restriction site. So it is also not suitable for recombinant DNA technology. Out of these enzymes, we have to take EcoR2 for the process of recombinant DNA technology. Okay, this is all about the enzymes. In the forthcoming video, we will discuss about vectors and the process. Okay, thank you all.